outside the German-speaking world, but he is the guy who not only said, hey, I don't think viruses exist, but also when he challenged people to prove it in court. Do viruses exist? And if so, they can win 100,000 euros, which is about $115,000, or 85,000 pounds.
We're talking about the famous measles trial.
Yeah. And nobody succeeded. There was one doctor backed by the whole of the pharmaceutical cartel, and even they were unable to present conclusive proof that viruses exist.
Yes. Dr. Lanka asked specifically for the one scientific paper proving the isolation of the measles virus and its diameter. And this German doctor sent him 6 papers claiming there's your virus.
Yeah.
And one of these papers is the reference paper with respect to the measles virus. So when you go on Wikipedia and you look for measles virus, it says measles virus was isolated in 1954 by John Franklin Anders. But the problem is that in his paper, there is no virus isolation according to the virology manuals, according to the classical method of virus isolation, which is called sucrose density gradient centrifugation. And every second-year biology student would be able, according to this method, to isolate a virus during one afternoon. But it's never been done. And in either this paper or in all other papers, what the scientists are doing, they are working with cell particles. Isolated cell particles from a cell culture, which is patented, which is kept alive, nota bene, which is kept alive only with the help of the so-called bovine fetal serum. This is a serum which is acellular and is obtained, mind me, sorry for this picture, but you have to get it to understand what is beyond the words. It's called bovine fetal serum. You find it in every vaccine. It's because the cells are only kept alive with this serum, which is obtained with a syringe by extracting this serum from the heart of an unborn calf in the body of the cow. And this is how they keep their cells alive in the lab, because all cells are dying in the lab. All cultured cells have nothing more in common with the cells in our body, with the tissues in our body. Everything is dying. Yeah, so they're doing experiments with these cells, claiming that because the cells die, there must be a virus that kills them. But what they don't do is they don't say how they prepare this cell culture. Yeah, which means, of course, first they take away all the nutrients which they had before given to the cells to keep them alive. They take away all the cells and they put antibiotics in the cell culture. So the cells would die anyway, no matter what you put in there afterwards, or if you don't put anything in there afterwards, if you just wait. Antibiotics kill the cells independently. There's no need for any fictitious virus. And they don't do control experiments, which of course it's crucial in science. You have